Ladies and gentlemen, welcome back to another video on the YouTube channel. So, now we are completely done with the anatomy of the skull, the bony anatomy of the skull. We're going to take a little bit of a break from the anatomy and move on to some histology, which is the subject that most students fear, despise, and hate with such passion because it is honestly a very tricky, difficult subject, especially with all the pink and blue and purple and all that to go. But I am here, so do not fear. I will try my best. I have tried my best, especially for today's topic, to make it as simple, as fun, and as easy as possible for you guys to understand the concept, see the images, and understand more than just blindly memorize. So let's get into it. I hope you guys enjoyed. This topic today is going to be about the epithelial cells, the basement membrane, and all that, or the histology of all of those. And yeah, let's just get into it. I hope you guys enjoy. Starting off with the epithelial cells. So as we know, this is just basic things. Bunch of cells make tissue. So we have epithelial cells and epithelial tissue. The epithelial tissue, which is the composition of multiple cells, lines the body cavities and the body surfaces. So if you go to imagine any body cavity, like your stomach, your intestine, your mouth, it's all, the first layer that's lined with is epithelial cells or epithelial tissue. Then what the other major presence is the body surfaces. The body surfaces, such as your skin, is lined by epithelial tissue. Logical. Now, because of their presence, or their location, where they're located, their function would differ. So the first function of epithelial cells would be to cover the lining, covering, lining, and protecting surfaces, which we said is in the epidermis. Here, this is a histological slide of the epidermal tissue, of the skin. And as you can see here, the major layer, the first layer, is the epithelial tissue. This is all epithelial tissue. Right, so it's okay if you guys are a bit confused on what you're seeing. There's a lot of things to process. Just focus on the topic of today, which is epithelial cells, which will be the outermost, not these little things, these little bad boys that are all cluttered together. These are the epithelial cells. That is what we want to focus on, just the dark purple color. Forget everything below, forget everything above, because every organelle or every location of the body has the, let's say, basic components are the same, but they also have their own add-ons, their own things that make them unique. So it's fine, just focus on the purple, which is the epithelial cells. Now, the second function is absorption. Absorption, such as in the intestines, like your large intestine, small intestine, all that is used for absorption. Over here, again, it's the dark purple. Forget those let's say flaying parts, those things that are jumping out like little projections. Forget those. We want the cells. These are the cells right here. This dark purple line, this is the epithelial cells. Just for you guys to understand, if you see any histological slide, you can immediately tell. That's why I'm putting different histological slides. Focus on what you want. Keep your eye on the ball. Nothing else matters. And one more function is the secretion, such as out of some specific glands. I did not put any histological slide for that because the glands are very complicated structure until you break them down, which I won't be doing, so it will be more confusing for you guys to see them now. So moving on now, we have the types of epithelial cells. Now, because they are so widespread, because they are everywhere in your body, you need specific types for specific regions. For example, my intestines and the palm of my hand cannot have the exact same type of cells. It's not logical. So, starting off with the cuboidal, from its name, it is cube-shaped cells. Now, under the light microscope, it's nearly impossible. You usually do not see the lipid bilayer. So you cannot exactly identify when a cell begins and when a cell ends. It's difficult to do that. It requires years of experience. So how do you get around that? How do you know that this cell is cuboidal, or if it's columnar, or if it's squamous? It all breaks down to the thing that you can see, which is the nucleus. See, look here, this dark stain. Here, obviously, we're using H&E stain. For those of you that want a video on the basics of histology, microscope, stains, etc., I'd gladly make it. Let me know in the comments and I will do it, no problem. But for now, let's just stick to what we have in front of us. Here, you can see the nucleus of these cells, correct? So the cuboidal cells, their nuclei, are always spherical and centered. See? They're perfect, almost perfect circles, spheres. You can tell from the nuclei what type of cell it is. That's it. Cuboidal cell, circular nuclei, done. Moving on. Next, we have the columnar cells. Now, the cuboidal cells are the cells that in shape are cuboidal. Columnar comes from the word column, which means they are elongated. Now, in this image, particularly in this image, you might think that the columnar and cuboidal cells nuclei look exactly the same. That is where you're wrong. The columnar cells most likely, usually, are more elongated. So in this image, it's not really clear that they're elongated. To get my point across, don't be stupid. Don't just memorize. Be smart about it. You don't need to know the shape of the nuclei to guess the cell. You can see the cell here perfectly because there's nothing above it to, let's say, disorient its borders. You can clearly see this right here. This is the end, and that's a column. You don't need to be like, oh, but the nucleus isn't circular or the nucleus isn't elongated. It is fine. You can see that it's a columnar cell. Keep it simple. Don't break it too much. Don't overdo it. Don't be over smart. Here you cannot, in this one, in the cuboidal, you cannot see the surface. You cannot see the exact lining of some cells. For example, here these linings are lost, but you can see the nuclei. So you can just say, okay, it's a cuboidal cell because of the shape of the nucleus. Here you can see the cell. Keep it columnar. Easy. Now the location of each of those I will get into whenever, like, where they are used. When we go through whatever part, whatever organ, I will tell you this is the cell, this is the image, etc. like that. Now, the squamous cell. The squamous cell is the cell that is known to have no definite shape. It can be any shape. It's not columnar, it's not cuboidal, it's not spherical. And in most squamous cells, as you can see here, the image is a bit too far off, but if you can see, the squamous cells nuclei are always flattened nuclei. So, whenever you want to identify any type of epithelium cell, first thing your eye should go to, the nucleus. Once you get that down, you know the exact type of cell. It is very simple. Yeah, that's it. That's all I have to say about the types of cells. Oh, one more thing. Sorry. Yeah, I knew I forgot something. The number. So, sometimes they can just be one layer, like in the columnar cell. Here, this is simple columnar because there's only one layer. They could be stratified, where there's multiple layers above each other. In that case, you need to know the number of nuclei so you can tell how many cells are present in that, because it's very hard to differentiate under light microscope the number of cells that are present above each other. Or they can be pseudo-stratified, where it looks like there's multiple cells but it's actually only one cell, and all that comes down to the nuclei. That's it. You just need to figure out the nucleus, what it looks like, and you will have it done. Now, epithelia itself, the epithelial cells or the epithelial tissue, does not have any blood vessels. It does not get supplied by any blood vessels. So how does it get its O2? How does it get its nutrients? All those are questions that are simple. Well, it has backup. Its backup happens to be known as the connective tissue. The connective tissue, my dear friends, supplies the epithelial. Oh, sorry, one more image on the whole thing that I just explained. The connective tissue helps supply the epithelial cells with O2 and nutrients. The connective tissue has a name. It is lamina propria. Lamina propria is filled with blood vessels, lymphatics, all those to help provide and clear waste from the epithelial cells. Now, if you want to talk about the directions, how in anatomy we have anterior and posterior, we also have specific names histologically. Anything above the epithelial cell. So let's say in this image right here, this here usually would be some sort of lumen or a space, either a lumen of a blood vessel, lumen of the intestine, or outside on the skin, that would be extra space. This is known as the apical pole. Got it? Anything above where the space is known as apical pole. APICAL pole. Anything below the epithelial cells, usually facing the basement membrane and the lamina propria, is known as the basal pole. Just the two poles you need to know. So just draw an imaginary line right here, apical pole, basal pole, and to help you apply it in specific cases, apical pole, basal pole. Simple. Basal comes from the word base. If you need some help memorizing basal pole, base, the base of something, which is the ground or the support of something, basal pole. base. Now, getting into this little thin line known as the basement membrane. The basement membrane, my dear friends, is the membrane, or let's say a structure that decides what goes from the lamina propria to the epithelial cells. So it is in other words, a semi-permeable filter for substances reaching the epithelial cells from below. Very basic, very simple. It also provides structural support, and it helps in repair and regeneration of the epithelial cells. Okay, now the basement membrane in itself, C here in this histological slide, C is the epithelial cells. Forget H, not important. C is the epithelial cells. BL, before I get to BL and RL, the basement membrane is split into two things: basal lamina and reticular lamina. Basal lamina, my dear friends, is BL. Reticular lamina is RL. Okay, basal lamina, reticular lamina. Now, just to help you guys understand why the image looks like this is not a light microscope. This is an electron microscope, because the basement membrane is so very thin. As you can see in this image, it's so very thin that it is really hard to break down and see. It is very small. You cannot see it. You cannot identify basal lamina and reticular lamina on light microscope, but you can on electron microscope, which is why this is here. Now, the basal lamina has macromolecules. The macromolecules inside the basement, or compose the basement lamina, are type 4 collagen, laminin, nidogen, and perlecan. Now, what is type 4 collagen? Type 4 collagen just helps in the stability. It is a network cross-linked together, 2D, for stability of the epithelial cells. Laminin are large glycoproteins that are attached to integrins. Integrins are small transmembrane proteins found inside the found on the basement or on the membrane of the epithelial cells that project through the collagen network. I will show you in a second. Stay with me. And nidogen and perlecan. Nidogen is a rod-like protein, and perlecan is a rod-like proteoglycan that cross-link laminin to collagen. They cross-link laminin, remember laminin, which is in the middle, to collagen. And they determine the porosity of the basement lamina, basal lamina. So, nidogen, perlecan are, let's say, assume they are what decides how big something can pass. So, they decide if small molecules can pass, large molecules can pass. It all determines on where the epithelial cell is present, on which organ is present, and the needs of that cell for that function. Now, laminin is just a large glycoprotein that attaches to integrins, and type 4 collagen is for stability. Now, reticular lamina, composed only of type 3 collagen. That's the composition of reticular lamina. It helps in support, and it is bound to the basal lamina by type 7 collagen. Is that clear? So, reticular lamina has type 3 collagen. It is bound to basal lamina by type 7 collagen. Basal lamina is composed of type 4 collagen, laminin, nidogen, and perlecan. And they are both rod-like structures that cross-link laminin. They cross-link laminin to collagen. So they connect laminin and collagen together, and they determine the porosity. So if they decide to put laminin and collagen close to each other, it would not be that porous. If they decide to put laminin and collagen farther from each other, it would be much more porous or much more permeable. Now, just to help you guys wrap your heads around this, because I know this image is very tough to get through and see, I painted an own image to try to help you guys understand. This is the epithelial cells, all right? The green dot, is the integrin. The integrin, which we said is a transmembranal protein present right here between the epithelial cells and the basal lamina, or the basement membrane. This forms the basement membrane. The basal lamina contains the type 4 collagen, laminin, nidogen. So laminin would connect to integrin, or attach to integrin, and projects through the collagen network. Alright, so laminin would project through here, attach here, and project through the network of collagen that will be present in the basal lamina. The reticular, and the last thing would be nidogen and proteoglycan, perlecan, which help in the cross-linking between the laminin and the type 4 collagen. Reticular lamina contains only type 3 collagen, and it is bound to the basal lamina by type 7 collagen, which produces its connection, helps in stability, helps securing each other, holding each other together so that nothing breaks and falls apart. And again, the lamina propria, which is the connective tissue that is filled with blood vessels and helps to supply the epithelial cells with oxygen, nutrients, and takes away the waste. That is it for the basement membrane for the epithelial cells. Next time, in our next video for histology, I will be describing the full of the gap junctions, the links between gap junctions and the proteins that link them together. It's a whole other thing. Hope you guys enjoyed. Thank you for watching. See you guys later.